All right. So first, we want to start with the subunits or the constituents of the protein, which is amino acid. And during my lecture, I'm going to use this abbreviation A.A. a lot. So whenever you see this, I'm talking about amino acids. So amino acids have different definitions in chemistry and biology. In chemistry, um, this amino acids basically includes any kinds of molecules that contain both amine group and the carboxyl group. So it doesn't necessarily have to be NH3 plus, as we've seen a lot in amino acids, but it can be anything with the functional groups, two functional groups, as well as hydrogen that are attached to nitrogen. But in the biochemistry, and not just the biochemistry, but all the biology, we actually refer to the specific configuration of the amino acids called the alpha amino acids. So when you look at the structure of amino acids that has a carbon here, right in the middle, that is attached with those two required functional groups, amino group and the carboxyl group, right? When these two groups are attached to the same carbon, this carbon is called as alpha carbon, and this is basically an alpha amino acids. But I mean, essentially, you can have multiple carbons that are linking to these two functional groups, right? So in case you have two carbons, you're always starting from the carbon that is right next to carbonic carboxyl group, and just name them as alpha, beta, gamma, delta, and so on. So in theory, you can have not just the beta amino acids, but gamma acids and delta acids, amino acids, and things like that. But again, for biochemistry and all biology, we just refer amino acids for this alpha amino acids. So this is a sort of a regular, template or the shape of the amino acids. As you know, there are two functional groups, amino group and the carboxyl group. And the other one is a hydrogen all the time. And there's a rest of one. So the alpha carbon has the highest valency here, which has the four bridges. And one of them is assigned for the functional group, or it's also called as R group. And there are, as many of you already know, there are 20 different types of amino acids. And basically, all these three components are the same, right? But except for this R group. So this R group actually gives the property or the characteristics of this amino acids, making in the different types of the molecules. So they are, naturally will vary in size too. The smallest amino acid is glycine, which has this hydrogen as its functional, the side chain or the R group, as opposed to tryptophan is the biggest amino acids that have a heterocyclic group for this R group. And basically this amino acid is a building block, right? So you're gonna have a polypeptide in the end, which consists of many amino acids that are linked together. And those each amino acids within the polypeptide are called as residues. All right. So again, I talked about the basically the building block or the subunit of the protein, which is amino acids. And these amino acids are linked together, especially by interacting between the carboxyl group and amino group, make a little chain like that, and then it can be extended basically because you're going to always have these two functional groups, the carboxyl group and amino group. And when you have several amino acids that are linked together, making a one nice thread, and then it's typically called as polypeptides. And these polypeptides will be extended even further to make some kind of functional entity called the protein. Let's talk about some basic properties of amino acids, especially in terms of chemistry and biochemistry. When you look at the structure of these amino acids, so here's our side chain or R group. It has the four legs basically, or four bridges, which makes the covalent bonding with the different functional groups. So naturally, it will have a heterohedral structure or the arrangement around this center, which is alpha carbon, and like we already discussed, the amino acids will have individuality, which is conferred by the diversity of this side chain or R group. And this, when you look at this alpha carbon, which is right in the center, this amino acid is chiral, and chiral meaning that it has handedness. So it can be either like the certain shape, like right-handed shape or left-handed shape. There are better terms for that, but we're gonna get to that during this lecture actually. But chiral or the chirality basically means that this molecule can form a structure, two different structures that are not superimposable on mirror images. And it basically applies to all different kinds of amino acids because they have all different kinds of functional groups that are attached to center, alpha carbon, except for glycine, which has hydrogen atom as the R group. So basically, it has two hydrogen atoms that are attached to that, which dissipates or the eliminates the chirality there. Question. Sorry, I didn't catch the later part. So. Not really. Can you speak up a little? I'm sorry. You should actually still keep the mask. I know. That's kind of. Oh, just what is it? It's the depth. So the question was that the alpha carbon is chiral, but how come it cannot be superimposed, right? Is that the question? The definition of chirality itself contains the not being able to superimpose on of the different configuration of the same molecule. So we're gonna get to that later on about the definitions of chirality as well as how these molecules can be superimposable or not be superimposed. So just wait for like I guess half an hour or so. But again, chirality is the same thing as have a property of the having the different molecular configurations that cannot be superimposable on the mirror images. These amino acids have different properties here too, which derived from its structure. So the amino acids is the zwitterions, and we already talked about what the zwitterions are, right? Which have the different charges within the same molecule, but the net charge is typically 0. That's the zwitterion, and that is due to the different ability to be ionized on the different parts of the same molecule, right? So when you look at this amino acids, Sure. So I was talking about the definition of a zwitterion, basically, which is a one molecule, but part of that molecule can be ionized, meaning that it can lose proton or accept the proton to have some charges, right? And depending on which part of the protein that, not the protein, molecule that you're looking at, they can be protonated or deprotonated having different charges, right? But the net charge is typically 0, meaning that one is positive, the other part is negative, things like that. It's not all the case, but at least the zwitterion is in that category. So, in which case, I mean, if you lose a polarity or not the polarity, but the charge from the one part of the molecule, then it's not going to be a zwitterion anymore. But it's an intermediate stage that becomes the zwitterion or something else, basically, like a neutral molecule. So, all right, make sense? So when you take into account of this amino acids, when you look at the two these functional groups, amino group and carboxyl group, Carboxyl group is pretty acidic, as you probably know, meaning that it can lose its hydrogen atom, which is called as a deprotonation, right? Or what is it? Hydro, not the hydration. What I'm looking for a slightly different word, but I it's just somehow it's just lose it. But will come back to me. But anyway, deprotonated can be deprotonated pretty easily. So it can become the COO minus here, which is a carboxyl group, as opposed to this amino group can be actually protonated, meaning that it accepts the hydrogen to become the positively charged. So it's going to be zitidirion in that case. And also this amino acid is amphoteric, and that means that this particular molecule can act as a base or also an acid, depending on what kind of partners for the reaction it is set up. So what would be the other example of amphoteric molecule? We already discussed one molecule during when we are talking about acid-base reactions. Water. So water molecule is amphoteric. Okay. Because it can work as a base, in certain cases, and also acid too. And you can imagine that since this is a zitidirion having a different properties, and also this, carboxyl group is acidic, and the amino group is more basic, it can, contribute to the reactions in a different ways in terms of acid-base reactions. Yeah, and keep talking about the properties of amino acids. Question. So the question was that how would that look like when you look at the amphoteric molecules like water or the amino acids? They're looking just completely normal, but they have a capacity to either lose the proton or accept the proton. That's the important feature that make them as a base or acid, right? So there are molecules. So imagine that you have many different functional groups there. One can work as a base, one can work as an acid, and things like that. And then you're going to have a higher chance to have a molecule, amphoteric molecule. Okay. All right. So let's keep discussing the properties of amino acids, and hopefully you remember this curve, which is a titration curve. And we already talked about this, that the most of amino acids, actually all of the amino acids, are having a capacity to release protons multiple times, right? And it's called as a polyprotic acid. So it releases proton multiple times. That's why it has the multiple titration curves throughout this titration event. And the y-axis is a pH, and x-axis is basically the hydroxide concentration, or the same thing as the hydrogen ions that are dissociated, right? Because when you're adding more and more hydroxide, you're actually depriving the more and more hydrogen cation. So it's the same thing there. And when you look at this amino acid here, which is actually, so here's a amino group, here's a carboxyl group, and H, one of these H should be another hydrogen on the bottom, and the last H should form the R group, right? So it's a glycine. And when you look at the glycine, and when it's set in the lowest pH, it has the plus charge because everything is not yet to be protonated. So it contains all the hydrogen or the hydrogen cations there. But as it goes through the titration curve, where you add more and more of hydroxide, or taking off the more and more of hydrogen cations, it'll lose the hydrogen cation one time here, which will have inflection point here. So there's a one titration curve. And then it goes through, and there's another one, another protein that is going to be removed from this amino group, right? Because there are two functional groups. There are two opportunities to be, the proton will be taken off. So that's why you have two curves here. And of course, depending on what kind of amino acids that you're looking at, it can happen 3 times or 4 times and things like that, right? Because of the R groups, whether it contains the one or multiple functional groups that can lose the protons. Make sense? So when you take into account in this titration curve, we always get the pKa value, right, which is an acid dissociation constant. And basically, the inflection point tells that the where the pH equals pKa. So that was one of the exam question, right? So there are going to be multiple inflection points in this case because this is polyprotic acid. And the smallest one, like a glycine, of course, will have 2 inflection points, so 2 pKa's. And you can actually get the pKa values, one from the carboxyl group and the other from the amino group here for the all the amino acids that are listed. Here it should be 20 there. And they're ranging about like 2.2 for the pK1, the first pKa value, and about 9.4 as an average for the second pKa value. But it's of course different from acid to amino acid to the another amino acids, right? And depending on what kind of side change you have, you're going to have another pKa value there. And let's just keep looking at the this glycine. So glycine's pK1 value is 2.34. But when you actually measure this number, you're going to see that this number is lower than what you expect by looking at this structure in many solvents. And the reason is because this carboxyl group in glycine have more opportunity to be close in vicinity to the amino group of another amino acid. So this amino group can stabilize the negative charge of this carboxyl group, so it can stay there without getting the proton and with a more like a lower internal energy. So again, there are at least the 2 titration curves with the 2 inflection points. And just hope you remember the Henderson-Hasselbalch equation where it makes the relationship between pH and pKa. All right, so let's keep discussing this pKa values for the polyprotic acid, the amino acids. So here's pK1, and here's pK2 for glycine. And then there's going to be a midpoint there, right, which is called as isoelectric point, or PI. And this is basically the amino acids where it has at the pH where it has no net charge. So net charge will be just 0. So imagine, on the following this titration curve of the glycine, first state would be most protonated form, right? So it has H in the carboxyl group as well as additional H in the amino group. So the net charge will be +1, like that. And then it just follows the this titration curve. There are more and more of the glycine that will become as a deuterium, where it loses 1 proton from the carboxyl group. So it's going to become like this. And there's 1 plus and 1 minus here. So the net charge will be 0, right? And there are going to be more and more of this configuration rather than this one as it goes to the higher pH or with the lower amount of hydrogen cation. And then it still kept going and then goes through the second inflection point where this molecule, with the net charge of 0, will become negatively charged, because now it also loses the proton from the amino group. So it's going to be neutral when you look at this amino group, not anymore like a +1 here. So the net charge will be -1, right? So simply the isoelectric point would be the right in the middle of this +1 or -1 charges. So that's going to be 0. And the calculation is pretty simple. Like I just explained, that's going to be an average of 2 pKa values, right? because the inflection point is right in the middle where the pH equals pKa. So you're just getting the midpoint right there. And the same thing can apply to the pretty much all the amino acids except for acidic amino acids and basic amino acids. Because acidic amino acids and basic amino acids have another charged groups in their side chains or the R groups. So you have to take into account of those pKa values too. So for the acidic amino acid, examples are aspartic acid or the glutamic acids that we're gonna talk about more in depth later this lecture. These guys have another carboxyl group on their side chains. So let's imagine that. Here's one example of the acidic amino acid, which is aspartic acid, D. And here's an amino group, and here's a carboxyl group, and here's a side chain also has a carboxyl group in it, right? And when you just count the total charge there, it's gonna be just +1, right? Because there's 1 plus charge from the amino acids. So that's the starting point. And when it goes through this titration process, one of them will lose proton, right? Become neutral. And then that's a neutral point. So that's gonna be the isoelectric point. And then it'll go even further out. Not that it's gonna be 0, right? Basically. And then this one will go through another titration curve with the inflection point where it loses another proton from the side chain. So that's gonna be -1, right? And then there's an amino group that will be eventually deprotonated again during the titration process. So the final stage would be with the charge of +2 instead of +1 here. Do you see that, the difference? Because you have another side chain that have capacity to release proton, you can actually have one more minus value, right? So it can go up to -2. And the point of getting the isoelectric point is just getting the midpoint, the net charge of 0, right? So that already happened from the first process where it loses the first proton from this pKa value, and the second one from this pKa value, nothing related with this amino group. So the calculation of the isoelectric point for the acidic amino acid would be using the first pKa value and also use the pKa value from side chain. Both are much more acidic than the amino group. And just getting the average of that. And basically, same thing can apply to the basic amino acids. So imagine you have one basic amino acid. The example is lysine here. Again, here's a carboxyl group. Here's an amino group, and this is a long chain of the functional group that ends with the another amino group, right? So in which case, this one is basic, and this one is basic too. So the starting point would be +2, right? Because both of them have capacity to accept protons. So instead of starting from +1, starting from +2, and then you have to basically lose 2 protons to get to an isoelectric point. So that's going to be, instead of using pK1, you're going to use pK2 and the pK side chain. Does that make sense to everybody? Because it can be confusing a little bit. And neutral is basically, an example is alanine here, which has the methyl group as its side chain or group. And simply, because it doesn't have any property, the R group doesn't have any property, it's going to be simply just getting an average of pK1 and pK2. So this PI, the isoelectric point, is really important actually for the biochemical assays and some kind of processes for testing the protein properties and things like that, because, especially for the protein purification, because the proteins have minimal solubility when pH equals PI, which is right in the middle here, right? And that means they're gonna be the most of the pool would be with the net charge of 0. having this form, right? In which case the protein will precipitate. Why? I mean, why does that happen? Why would protein precipitate when the pH equals Pi? Can anyone answer to this question? So they're not, because they're not ionized. Are they ionized or not ionized? They are the most ionized form because they have both positive charge and negative charge. And that's the definition of the Zephyr-Ryan, right? They're partially ionized everywhere. So it's basically the most ionized form compared to this, imagine this. There's only one that is ionized. But it'll precipitate, basically. It will have minimal solubility to water, especially. How come? It could. It definitely can. Because that is correct. And would that matter? I mean, the fact that it doesn't have any charge, is that important in terms of the solubility to water? How come? Exactly. So water molecule itself is polar, right? So it likes polar molecules. And charged molecules like ions are the best one, right? For the water, for solubility. And of course, at the isoelectric point, the net charge is 0. So it's kind of neutral there, sitting there. So it's not hard to interact with the water molecules, which is polar. Make sense? All right, so, well, unfortunately, we have 20 amino acids in the proteins instead of, 4 nucleotides in DNA, and which means that there are more things to memorize, I guess. I'm sorry. I'm not enjoying this, but at least there are things, these are really critical, so we should be well aware of their structures and should be able to distinguish them by looking at especially R groups. So we can group those amino acids in many different ways, but one common way to do it is to look at their charges. So we have 9 non-polar amino acids, 6 polar but uncharged amino acids, and 3 basic and 2 acidic amino acids that forms the all 20 amino acids. And you can see their structures and their properties and their functional groups and things like that basically in the textbook in page 82 and 83 if you're using the same version as I do. Here are some examples of the different amino acids in different groups. The first one are the amino acids with a non-polar side chains. So this is the first category here, which basically has the hydrocarbons as their R group or side chains. So like CH3 or CH, 2H, C2H6 and things like that, kind of simple kind of structure there, which does not have any polarity or the charges and things like that. And also it can contain the phenyl ring like this. And another example of a grouping is ones with the uncharged polar side chains. So uncharged meaning that they're not ionized, they don't have any charge, plus or minus, but they still have a polarity there that can be set up by the basically dipole moment. And these groups contain like hydroxyl group or sulfhydryl group and things like that. And also another example is ones with the charged polar side chains. Charged meaning that they're ionized, right? So they're either acidic or basic. So the examples are here too. And also there are different groupings like aromatic amino acids, which are tryptophan, phenylalanine, tyrosine, and also sulfur containing amino acids like cysteine and methionine there. And majority of globular proteins in there because they are sort of soluble there. The globular protein, meaning that they're sort of floating in the cytoplasm or also inside the nucleus and things like that, kind of having interaction with the other things time to time, but typically just sit there, just floating there, will have the non-polar amino acids inside of that, and the polar or the charged amino acid outside of the protein structure, because it has to interact with the water molecule. It's all about using the water molecule as a solvent, right? Because it's our like condition, the cellular environment. There are 3-letter codes like glycine, leucine, isoleucine, threonine, and things like that. And also, there are more conveniently have the first just the letter, 1-letter codes, and typically they use the first letters for the G, L, I, T for those amino acids that I talked, and also the second letter can be used in case of first letter is already taken, like tyrosine and arginine and so on. And if there's nothing really available there, and then we use some mnemonic rules like aspartic acid, D for aspartic acid, Q for glutamic acid, kind of similar to G, I guess. Phenylalanine is written as F, and asparagine, and so on. So again, go to this table in the textbook, then you're going to get all the information about these. Now we're going to go over these different groupings of the amino acids, and we'll study their properties, biochemical properties. First group question. That is a good question. So the question was basically that you just look at a single protein there, and protein consists of a lot of different amino acids. Some of them are acidic, some of them are basic, some of them are polar, non-polar, and stuff like that. How would you be able to calculate the isoelectric point there? It's just additive, basically. So you just take into account of all the single amino acids all together, and just calculate them. It's just getting the mean of all the pKa values. But if you're seeing the acidic amino acids, then that's going to be this equation. If basic, this equation, and the neutral ones will be this equation, and just adding all of them up, and then divide by the number of pKa's that you can see. So getting an average value. That's going to be an isoelectric point for that protein. And it's not perfect, right? It's not perfect because partially it's positively charged, partially negative charges, and things like that. But overall, it's going to have a net charge there, have a minimum solubility to water. So again, you can use that property for precipitating the certain protein. And precipitating is a good thing because you have a sedimentation there. So you can simply just get rid of the supernatant of that solution to get that pure protein. Of course, there are different techniques, a lot of techniques to help this out further to purify those proteins that we're going to discuss much further out. But again, this is a really good property and a good starting point for protein purification. Okay. So the first grouping is amino acids with aliphatic side chains, and aliphatic meaning that it consists of hydrocarbons, so it does not have a polar, right? Like alkane, and mostly the side chains with the amino acids with the aliphatic side chains have alkane structure, meaning that all of them are single bonds, single covalent bondings. The examples are glycine, alanine, valine, leucine, isoleucine here, and basically this is listed or placed where having the smallest R group to the largest R group here. And as you can see, these are just a pretty simple, like just a hydrogen or the hydrocarbon, like methyl group, isopropyl group, isobutyl group in a different configuration and things like that. As it moves from glycine to isoleucine, it'll be more hydrophobic. And you can imagine that, right? Because it basically have a bulkier non-polar side chain. So it's going to be less polar or less, hydrophilic. And that means that these guys here, which have a bulkier hydrocarbon R groups, will have a better solubility to hydrocarbon solvents like benzene or petroleum and so on. And these guys, the hydrophobic molecules, the amino acids, are typically buried inside the protein molecule, again, to minimize interaction with the water molecule, which is polar, in which case it'll raise the internal energy and make the everything pretty unstable, right? All right, so the next grouping is the amino acids with the hydroxyl or sulfur-containing R groups. And these four are the amino acids within that category, which are serine, cysteine, threonine, and the methionine. I'm sorry, I was just blinking a little bit. And these serines and threonines are really biologically important and significant amino acids because when you look at them, it contains a hydroxyl group in their side chains. And these hydroxyl groups are a really good substrate for phosphorylation, meaning that adding the phosphate group there. And this actually is really important post-translational regulate protein function and localization regulation mechanism. So we're not going to discuss too much about this function of the phosphorylation because this is biochemistry, but hopefully you can cover a lot of things in other fields like cell biology or molecular biology. So these amino acids that containing hydroxyl or sulfur containing R groups are not going to be ionized easily in the under physiological condition like a pH 6 to 8. For example, the cysteine ionizes only at pH 8.5 to 9.0, meaning that does that mean this is, so when you look at it, there are hydroxyl group or sulfide group, meaning that it can actually release protons there, right? It can, in theory, but it seems like it doesn't do that. No ionization at pH 7, meaning that it doesn't release proton at the physiological pH. And that means, is that a weak acid or strong acid? Weak or strong? Weak. That means a weak acid, right? So it try to just hold on to that proton, doesn't want to release it even at pH 7 or so. If it goes higher, where there are more of hydroxide there, less of hydrogen, then now for the equilibrium, it will release the proton there. So that's why they're not called as acidic amino acids, okay? All right, so, and they have the weak polar R groups, of course, weak acids, so weakly polar. And they're generally a little bit more hydrophilic than aliphatic amino acids at a comparable size because you can see the polarity there that is built up by the hydroxyl group or sulfide group, except for methionine, which resembles the hydrocarbon, right? Except for this sulfur here. So that's why it's quite more hydrophobic. And cysteine is really interesting and also really important for the building up the protein structure because it builds the disulfide bonds, having the covalent bond between two sulfurs within or between polypeptides that we're going to also talk about in a little bit later. Another group is aromatic amino acids, and these three amino acids are within that category: phenylalanine, tyrosine, and tryptophan. And as you can see clearly, they contain either phenyl ring or the fused, some kind of carbon rings there. And this, especially this tryptophan, contains the nitrogen here, which also confers an interesting property there too. So when you look at the first amino acid, the phenylalanine, it is one of the most hydrophobic amino acids because there's not much of a polarity can be built up there other than just this phenyl ring, which is really hydrophobic and really non-polar, right? There's no directionality or charges or anything there. So again, phenylalanine is the most hydrophobic amino acid. As opposed to phenylalanine, the tyrosines and tryptophan are relatively a little bit more hydrophilic. I shouldn't say hydrophilic, but less hydrophobic because it has another functional groups that are attached there, like a hydroxyl group, or this kind of fused rings that make a little bit of a polarity there. And when you look at this structure of the R group of the tryptophan, it actually resembles a little bit of the purine, right, in your DNA. Except for missing some of the nitrogens here and there, a couple of nitrogens. And that means that it can actually set up a little bit of a polarity there. And that is really interesting because it actually exhibits a similar property as the purines. It can provide the stacking force too between the different amino acid, the tryptophans within the protein. Again, have the sort of a van der Waals force set up there, right? So this one, and that makes the dipole moments there and the sort of the making the structure a little bit stabilized in the polar form, so it can be a little bit hydrophilic. As opposed to this one's still kind of sort of in the middle of this phenylalanine and tryptophan. And the last grouping here would be the acidic amino acids, basic amino acids, as well as amides for the acidic acids. So they are definitely hydrophilic because they are charged, right? Take a look at the structure of these amino acids here. One is aspartic acid here, and the glutamic acids are here. And obviously, they are acids because they have contained the carboxyl groups in their side chains. So can easily release a proton there. And the asparagine and glutamines are another examples of the hydrophilic amino acids. I'm going to just skip over to this basic amino acids. There are 3 of them: histidine, lysine, and arginine. And all of them, as you can see, have some kind of nitrogen atom that is involved there, making imidazole structure, or just having this amino group, or the guanidino group, which contains, when you look at it more closely, it contains this amide kind of structure here. And they're usually found in the surface of the protein because they are polar, right? So they like to interact with water molecule. So of course they're going to be exposed in most cases to aqueous environment. But also they can be located inside the protein, so inside the folded protein, because it can still make the hydrogen bonding or interact with the other regions of the protein using van der Waals force. Especially the polypeptide backbones here. When you look at them, so this is a polypeptide, and there are peptide bondings, here and here and here, and things like that, and so on. And there are side chains here. If you have side chain with the acidic property, then this one can interact with the backbones, which are basically right in the middle here, and NH can be work as an acceptor because the hydrogen atom is attached to the electronegative atom, which is nitrogen. And also here, the CO, which is ketone, basically. This oxygen can be, work as an acceptor for the hydrogen bonding. So there are a lot of places where the hydrogen bonding can occur, right? With the side chains or even water molecules. And also because they have charges, they contribute to the overall charge of the protein, whether it's going to be positive or negative. Here, I mean, this is pretty clear that the acidic amino acids as well as basic amino acids have charges and hydrophilic. But what about these asparagine and glutamines? They do not have any charges there. They're just amides. Amides meaning that there's a CO, N, and something, functional groups that are attached. When you look at it, CO and N, and instead of 2 R's, there's H2, right? So this amide group, functional group is attached there. But this one also is hydrophilic because it can build the polarity. As you can see, this is not like simple hydrocarbon structure, right? So using the van der Waals force, still it can interact with water molecules or something else that are polar. Here's a quick summary in the middle. We talked about all the hydrophobic amino acids like this, and some of them are just containing the hydrocarbons at their side chain, and some of them contain the aromatic rings in their side chains, and they're insoluble in water or tend not to be solubilized in water, and also they tend to be inside the protein, making the sort of a cluster. But in contrast, the hydrophilic amino acids, which are listed here, are the polar, basic, and acidic amino acids that have charges or have a polarity there, are easily ionized even at the pH of 7 because they're acids or bases, and typically containing the amide or hydroxyl group or sulfide group and so on. And these R groups are participating in H bonding, interacting with the other polar groups of the amino acids or the water molecules or even the backbone of the polypeptide, and usually found on the surface of the soluble protein or globular protein, and can also be located inside the protein, but will have H bonding with the polypeptide backbone. We haven't talked too much about other three amino acids. Here, one's glycine, the other is proline, and the last one is a cysteine. So we're going to talk about these two amino acids from now on. The glycine is the smallest one and has the hydrogen atom as its R group or side chain. So since it has two H's here, it's not chiral, meaning that this is not having the different configurations that are not superimposable on the mirror images, right? So there are basically there's a symmetry around this alpha carbon. Again, this is the smallest, and that actually means that it's a versatile amino acids because it can be inserted anywhere. It's small, it's neutral, so it can be inserted anywhere there whenever there's a little space there without affecting too much of the charges or interactions with the other amino acids and things like that. And of course, it's smallest, so it will have least steric hindrance or steric clashes, and can readily fit into any kinds of structural elements like some kind of helix or some kind of sheet or some kind of barrels and things like that, except for actually alpha helices, which make those kind of spring-like, metal spring-like a shape, because it has no properties. And to hold that alpha helical structure, there has to be some interaction between the amino acids within that peptide. Just like a DNA helical structure, right? There has to be a base pairing there to hold those structures together. So since glycine doesn't have any property there, it will actually interrupt the polypeptide forming the alpha helical structure. But instead, it can be mostly used in turns and loops of the protein. Cysteine is the next one, and interestingly, it has the sulfhydryl group at the end of its side chain, and it can be basically forming the covalent bonding. I don't know. Sometimes I'm just thinking today. Covalent bonding with the other cysteine to make this kind of 2-cysteine molecule, and it's also called as a cysteine. So it's just the same pronunciation, which is kind of, I don't know, unlucky thing. Yes? That's a really good question. So the question was that the methionine contains sulfur there too, right? So can it just make another like disulfide bond with the cysteine or some other methionine? But the answer is no, because its the sulfur is right in the middle of that, sort of a hydrocarbon-like shape. So it has no, places, no valency to interact with the other, molecules or the other functional groups. As opposed to this one, the sulfhydryl group is exposed to the outermost side, region of the amino acid. So it can actually interact it. And also it has capacity to release this proton, right? So that's another important features. So cysteine is the only amino acid that can make the disulfide bond. And it is really important because this disulfide bond is really strong. Covalent bonding can happen intramolecularly as well as intermolecularly to form the ternary structure of the protein. And the last one, that's the interesting amino acid was the proline, and it's technically amino acid, not an amino acid. And imine is the here's the structure of imine, where it resembles amino group, but it lacks the high, what is it, the high the oxygen atom in the this the peptide structure. I don't know what's up with me today. And the this proline is the only cyclic amino acid. When you look at the structure here, it is different from the other amino acids, right? Instead of having this accessible amino group exposed, it actually is linked together to the side chain like that. So here's another indication here that you can see that this side chain, supposedly side chain, is going back, looping back to the this amino group to form the covalent bonding there. So it's a cyclic structure formed there. And this R group is, as you can see, aliphatic because all the carbons and hydrogens are forming this side chain. And since this side chain is linked to the amino group of the proline, this proline residue is relatively, the molecule itself is relatively less flexible, especially in the peptide chain. You can imagine that, right? There's no flexibility built up there because there are two covalent bonding that are attached to the amino group. So it's kind of just stuck there and will not allow any kinds of rotational movements too much. And that will actually make the certain region of the protein more rigid. And that is sometimes really important, especially for making some kind of functional structures there, or kind of interface, designing the interface that can interact with the particular other protein partners. So these proline residues are not easily folded into protein structure because of this rigid structure. So instead of included in the some kind of repetitive structure, it is located in the places where this polypeptides make some kind of kink, like abrupt kink. So it's often called as the alpha helix breaker, because they're going to be alpha helix, and then whenever there's a proline, they're going to be a kink, and then there's going to be another structure that starts. And so they're going to be mostly, the prolines are going to be mostly included in turns or loops, just like glycine, but will be more rigid, much more rigid. So it's going to have a stronger structure there. So far, we talked about all the different properties of amino acids, and we went over basically all the amino acids, their, how they structured, and what it means, the functional groups, and how they interact, and where they're localized, and things like that. All these amino acids have a chirality, except for glycine, right? So we want to talk a little bit more about this chirality, property of being chiral, and that means that it can have handedness, or that it can have its match, the mirror images that are not superimposable. These chiral compounds are always optically active, meaning that there's some kind of optical measures gets in there, then will be active, meaning that will respond in a different way, certain way. And basically, what it does is that you have light source, and then you can make the polarized light going through this polarizer, it can be any directions there. And so you're going to have plain polarized lights there. And when you lit it to the any kinds of compounds that are chiral, have the chirality, then since it has a chirality, it'll be oriented. All the molecules will be oriented in a certain way. So there's going to be a certain like a consistent diffraction will occur of that light, will turn or rotate this plane of phase of the light. Then you can actually collect that how much of that by looking at it through the other polarizer. It can be either look like a completely bright or some kind of dim, and depending on the brightness that you can see, you can tell that how much of this polarized light was rotated. And the molecules have chirality are the called as enantiomers. So we want to start with this is enantiomer is a subgroup of isomers, and we already talked about isomers, right? Basically, it's the definitions right here. It's the molecules that share the same compounds, the same constituents. But when they have the same constituents but have a different connectivity, different orientation of the molecules, we called it as constitutional isomers, right? We talked about that, and the example of constitutional isomer is tautomer. We talked about the tautomerization of the nucleic acid, the nucleotides that can be keto form or enol form. So those are the constitutional isomers. And there are different categories under these isomers, which is called stereoisomers. And enantiomers are one of them. And stereoisomers are basically still have the same formula, meaning that they have same constituents, and also have the same connectivity, but they have different arrangement of those molecules. So they are connected on the same places, but one can place, towards you or the back of this kind of like towards back or the up and down and things like that. They're sort of arrangement, the orientation can be different, and those molecules are stereoisomers basically. And enantiomers are even further out that those like stereoisomers in that have mirror images, so they cannot be superimposed. An example of this molecule, small molecule, is right here. So they have all different colors there, right? Even if you turn this molecule 180 degree to align the red and blue and green molecules together, you're going to see that this light green ones are either going towards you or the back of the plane of this basically presentation here. So you're not going to be able to superimpose in any cases, and that's called as enantiomers. As opposed to there are cases where they are stereoisomers, but also does not make the mirror images. They're called the diastereomers. And this enantiomers have basically identical chemical, physical properties because they have all the same constituents, and their shape and arrangement are pretty much the same, except for that they have like opposite of the mirror images, right? The only properties in that they are different in most cases is the activity, the optical activity, where, like I told you in the previous slide, it can actually, the enantiomers or any chiral molecules can rotate the polarized light through the column. But that polarization will happen in the opposite direction between the different enantiomers because their arrangements are basically in the opposite direction, right? Like mirror images. But they're going to be having the consistent structure or the organization there. So we'll turn it in the same degree though. And the racemic mixture is basically same amount, the 50-50 of the enantiomers. So in which case, it's not going to be optically active, meaning that even if you lit the polarized light, you're not going to see any rotation of that polarized light phase, because there's exactly 50% of the different enantiomers that are mixed together really well. So it's going to turn, halfway right end, halfway left end, and things like that. But overall, it's going to be the same. And interestingly, in the cell, we only see L enantiomers, a specific configuration of enantiomers of amino acids that we're going to talk about a little later. But you can actually generate both D and L form, like both mirror images. But the fact that you can only see the mostly L form of enantiomers of amino acids, telling that it probably have some kind of selectivity there, especially for the enzymes that are involved in synthesizing those amino acids. So to tell that which enantiomers that we're distinguishing and talking about, to make things consistent and easy, we use this Fischer-Ransonov convention, or DL system, and D is for Dexter in Greek, and L Latin, and L is for Levis, so right-handed or left-handed basically, and they can tell the what kind of optical isomers that they have. And they use basically this Fischer proposed to use the molecular configuration of the glyceraldehyde, which has a pretty simple structure, but have all different kinds of functional groups that are attached to the chiral center. So chiral center, which is the carbon or some kind of atom that is located right in the middle for them making that chiral molecule. And simply, you just put the chiral center in the middle, and locate the hydrogen, the smallest atom on the right-hand side, and put this aldehyde group and this acetone group on top and bottom, and then you can basically rotate, put this hydrogen and hydroxyl group in the different ways, like whether this one, the hydroxyl group especially, goes right-hand side or the left-hand side. You can basically tell that this is D form or the L form. So if the hydroxyl group goes left side, then it's going to be called as Levis L form, which is left. And if the hydroxyl group is located on the right side, that's going to be D form. And this Fischer projection is also convenient because you don't need to write this kind of arrowhead kind of structure anymore, because it just assumes that you just put all the molecules in the same way to observe it, where these two horizontal bars are the molecules are going to be above of this plane that you're looking at and in the projector, and these two other molecules that are located vertically are going to be behind of this chiral center. So we can apply the same DL conventions for amino acids too. Put it in the similar way here. So H is in the same place here, and we're looking at the location of this amino group. And there's a carboxyl group that we matches with the aldehyde group here, and the R group, the biggest one, will be located as the same way as the acetone group in the glyceraldehyde. And basically where you can see where this amino acid amino group, it can tell that you can tell whether it's a L form, which is on the left, or the D form, which is on the right. And there's a different convention also kind of make things easier to count the test that whether this amino acid is in D form or L form. It's called the Korn rule, where you place this chiral center in the middle, and place the hydrogen atom at the back of the chiral center, and you can see that how these other functional groups are oriented or arranged. If you can see that the carboxyl group CO and the functional group, which is methyl here, because it's alanine, and the NH2, the amino group that goes clockwise, then that's going to be D form. If that is counterclockwise, then that's going to be L form. There are 20 standard amino acids, and there are many of them are or all of them are used for the protein biosynthesis, basically, translation, making the proteins. And organisms are differ in the how many of these amino acids are produced and utilized. And there are also some other amino acids called essential amino acids that have to be obtained from your food, basically your diet. But the rest of them can be biosynthesized and from the much simpler molecules. And there's a general genetic code that are used to basically convert the energy, information from the DNA to protein, which is called as codon. And this codon consists of 3 consecutive nucleotides or the bases in the DNA, and that actually encodes one residue, one amino acid in the polypeptide. And that basically has the linearity of the DNA and RNA and protein, and also determines the primary structure of the protein. So speaking of the codon, it's basically the rule for getting the genetic information and converting it into proteins, which are actually actual working entities in the cell, and that means the information from the DNA or RNA is converted to the amino acid sequences. And again, there's a linearity following this genetic code or codon rules. And this is the table that basically listed out all the relationship between the codon and the amino acids. So for example, if you read it as AGU as a codon in your mRNA, you can just find it from first base of the codon here, A, and then G, and then U, third one's right here, which is going to be serine right here, right? Same thing applies to the all other molecules. And this is not just happening simply, spontaneously, but it involves this tRNAs. And tRNA, you can just imagine its structure as this kind of clover kind of shape, but it actually have a different particular kind of 3D structure, which looks more like the kind of wedge with a little like tail here. And we just talked about the essential amino acids. There are 10 essential amino acids, and essential amino acids are basically something that you have to get from your diet. So the body cannot generate this from any other different other small molecules. So these are the list. This is the list of the essential amino acids here, and especially for histidine and arginine are considered as essential amino acids only in your childhood, because during the processing, the enzymes and the biochemical processes for generating these 2 amino acids are not well developed, so you still have to get them from your diet. But once you get to adulthood, then basically the body can generate these 2 amino acids too. And there are also non-standard amino acids. There are about 900 amino acids that are produced by natural pathways. So there are a lot of them, right? But the living organisms typically use only 22 of these amino acids to form the proteins. So there are 22 proteinogenic amino acids, which includes 20 standard amino acids that we talked about so far. And there are 2 more non-standard amino acids, which are selenocysteine and pyrrolysine. And selenocysteine is basically having the same structure as a cysteine or serine, but instead of having oxygen or sulfur atom, it has a selenium. So that's why it's called as a selenocysteine. And pyrrolysine, as its name says, pyro, it's the fuse of 2 lysine molecules. So it's looks like this kind of form. They are really rare in proteins, but still really important, forming certain enzymes and things like that. And also, it can be encoded from the DNA using the genetic code, which is a UAG. And this UAG is typically a stop codon. So in many cases, instead of generating this selenocysteine or pyrolysins, it's going to just stop the translation and finish it and make it into a protein. But in some rare cases, there are going to be other mechanisms involved there to use this codon for inserting these amino acids. And for making these amino acids as a polymer, there should be a process of fusing them together or making the condensation. It's called the peptide bond and peptide bond formation, which is a part of a amide bond. And amide bond is more like a bigger term where whenever there's a reaction happens between amine and the carboxyl acid and forming some kind of structure there, it's going to be called as amide bond that makes the amide. But the peptide bond is only used, the term that is used for alpha amino acids in biochemistry, where it makes this kind of polymers for the making the polypeptides or the protein. And again, goes through this process where the carboxyl group is interacting with the amino group, where the water molecule goes basically is formed and taken out of these two molecules to form this peptide bond. So this process is called as dehydration condensation. And typically these process, peptide bond formation, forms the linear structure of amino acids, multiple amino acids. So the polypeptide will be mostly linear, but sometimes the cysteine bonding, the disulfide bond between the two cysteine residues can form give them some kind of other structures, so make them some kind of branches on the polypeptides and so on. And this polymerization of amino acids or translation makes the protein, and water molecule is a leaving group of this process. And it doesn't just occur by itself again, but it requires two things. One is in 5 minutes, one is the amino acid tRNA. So tRNA that is fused to the amino acid, and also the catalyst called the ribosome. It's an enzyme that basically matches this particular amino acid tRNAs to the codon, and then will make the polymer of the polypeptides, the protein, basically. And that is linked, those amino acids are linked by sorry about that, tRNA and the amino acids are linked by the ester bond using ATP. And this protein synthesis, basically, polypeptide synthesis, always happens from N terminus to C terminus, just like the DNA always synthesizes from 5' to 3'. And proteins, in the end, proteins are important because they're the one actually do all the work, right? So they can be work as catalysts, scaffolds for some kind of higher order structures, transporters, regulators, and so on, basically. And regulators, like they're involved in some kind of cell signaling pathways, and then convert certain molecules or induce the certain transcriptional activation pathway and so on. And these proteins are the most important macromolecules because they're consisting of more than 50% of dry weight. So there are a lot of proteins there, always synthesized and degraded. And there are many, many exotic proteins that are really useful, particularly in the lab. Examples are GFPs, green fluorescent proteins from jellyfish, luciferase from firefly, antifreeze, reagents or the solution for the Arctic fish, and the honeybee venom malatin is used for the arthritis treatment. And you just observe this interesting exotic event, and then get to the mechanism how it occurs, then purify those proteins and engineer them, and finally use into your system of study to utilize that to look at the certain like new features. And it also, proteins have a really high diversity because it forms with the 20 different amino acids as building blocks. So you can imagine that there can be a lot of different combinations exist. It's much beyond to the number of particles in the universe that are calculated. So this is the last slide. There are also atypical peptide bond formation exists in the cellular system, and they are synthesized basically from the regular amino acids, but using the specific enzymes they can form this specific interesting ones. One example is a tripeptide glutathione. And that has a really plays an important role because it works as an antioxidant. And as you know, this is important for anti-aging, right? And that basically protects the cell against the oxidative stress, like the hypoxia. It can be hypoxia, or the attacking from the excess of the reactive oxygen species like ROS. And the synthesizer step is actually pretty simple. So it goes through the two steps. First step would be the glutamine and cysteine get condensed by using the enzyme called gamma glutamyl cysteine synthase. And what it produces is a glutamyl cysteine here, which is here too through the pathway. And it consumes the ATP here. And the next one would be this glutamyl cysteine can be condensed down, fused to the glycine using this glutathione synthase or GS here. And then this one can be delivered to the many different organelles like nuclei, ERs, mitochondria, and so on to protect the cells from the oxidative stress. All right. I have one more thing, but we're going to discuss this next time. So